Hello everyone, and welcome to a short presentation on qPCR testing. Just to mention, qPCR testing is available through Pace Energy Services in Pittsburgh, Pennsylvania. So, the big picture. The big picture is that microbes degrade contaminants. There's a lot that goes into this, but the bottom line is that microbes convert toxic chemicals to carbon dioxide, water, and salt. So what's qPCR? qPCR is a way to find out how many contaminant-degrading microbes are at a site, and it comes in two different flavors. The first is 16S, which is a region of the bacterial DNA that tells you who it is. Second are functional genes. Functional genes essentially tell you what degradation activity is possible. So with qPCR testing, you can get who and what. So what kinds of samples? Well, we can analyze just about any type of sample. For surface or groundwater, Pace Energy will supply a filter that can be used with just about any type of a pump. We like to see about 1 liter or so of water pass through the filter, but we can also analyze just about any type of porous media like soil, sediment, even rock. Also, any type of engineered coupons that folks may use, for downwell experiments or whatnot. Also, there's a short hold time on qPCR, so the sooner that you get it to the lab, the better. Just some notes on qPCR workflow. The lab receives a sample, and there's some QC steps to do, and then the DNA is extracted and purified. Once that's done, there's another QC step. Then the samples are loaded onto the instrument and run. Once that's done, there's more QC checks, and if all that's okay, a report is then generated. This is what the data looks like from the qPCR instrument. You see on the left, as the DNA is amplified through thermocycling, there's a resulting fluorescent signal that can be quantified based on a calibration curve of that target. The calibration curve is on the right. So this can be done for any particular microbe using, again, 16S, the 16S gene, or any gene, any functional gene. So here's what a report looks like. It has all the pertinent information. And as for the report, in this one right here, there was no detection of the target bacteria in well 29S, but there was a detection, 1.47 times 10 to the 4, or 14,700 gene copies per mil in well MW29D. So as far as taking samples, as a lab, we'd like you to take samples everywhere all the time. But obviously, where you take samples depends on the site objectives and goals of the particular project. But usually you don't need to analyze every well. A good rule of thumb is to sample where things change. Now that change could be geochemical, contaminant levels, or something else. So here's the money shot. Sites go through stages, and qPCR can provide useful and actionable information at every stage. Some of the questions that can be answered include, are contaminant-degrading organisms present? If so, how many? Is monitored natural attenuation feasible? Are amendments necessary? Is bioaugmentation necessary? Why are contaminant intermediates accumulating? Why is that cis accumulating? Did the amendment promote growth of contaminant degraders? Did the inoculum survive? Is it being maintained? Is the population stable? So these are the types of questions that can be both asked and answered. Here's the cost. At this time, we have qPCR targets focused on reductive dechlorination, but we're adding new targets about every 2 to 3 weeks. Mention you saw this presentation, and we'll give you a special discount. If you have any questions about qPCR or any of the other tests that we do, please reach out to your local PACE account executive or give us a call. Thanks for watching.